A very good afternoon and welcome you all back to the afternoon session on day one of training on pilot plant scale-up techniques. So now I take the privilege to introduce party. So Sar has completed his awarded with his PhD in pharmaceutical sciences from Andhra University in the year 2013 under the expert guidance of Dr. Y. Rajendra Prasad Gar. He was awarded with CSIR Senior Research Fellowship by Government of India for completing his PhD degree. Sar is also a registered pharmacist, young scientist, and an expert in the field of design and synthesis of bioactive hybrid molecules, computational drug discovery with a focus on pioneering of hit-to-lead optimizations associated with the development and novel bioactive ligands to fight against multifactorial diseases. Few of his achievements include he was recipient of the International Medical University E-Learning Achievement Award 2019, and also SAR is an innovator on technology enhanced learning strategies and a principal investigator of the prestigious FRGS Grant 2019, which was awarded by the Ministry of Education, Malaysia. He is an International Medical University Learning Resource Festive University Competition Gold Medal winner also. He has showcased his project in the title Virtual Innovations via Simulation Training and Assessment. In the second Higher Education Leadership Meet 2019, the Venus International Foundation recognized and conferred him as Young Leader in Health and Medical Sciences discipline. Dr. Vasudeva Rao is currently a senior lecturer in Pharmaceutical Chemistry Department and an e-learning lead in School of Pharmacy at the International Medical University, Malaysia. He developed innovative teaching and learning methodologies for the various course modules based on new drug discovery in undergraduate and postgraduate programs at IMU, which help students to practice theories and principles of drug discovery and development. So with this brief introduction, now I request Dr. Vasudeva Rao sir to kindly proceed with the session. Over to you, sir.
Thank you so much, Dr. Pramod, for the well introduction. And firstly, I would like to thank International Medical University, Kuala Lumpur, Malaysia, for granting me the approval to participate in prestigious event organized by Jabrul Hanumai Institute of Pharmaceutical Sciences in collaboration with AICTE and ISTE. So, dear participants, welcome to you all to this introduction, the refreshment program. So without any further ado, I'm going to share my slides and discuss about the topic of today's section. May I have the confirmation? Is that everyone can see my slides?
Yes, sir. The slides are visible.
Okay, thank you so much. So, today our topic of discussion is simulation integrated scaling of processes in drug discovery from bench to market. My name is Dr. Vasudeva Rao. I'm from Kuala Lumpur, Malaysia. So just I would like to give a small update to you all. And yesterday's cases reported around 0.24 million cases. So still we are in the pandemic situation. So please all of you stay safe and take care. So what's the aim of today's webinar presentation? The main aim of this webinar is to introduce participants with the information related to the role of simulation for scaling of processes in drug discovery. Moving forward, upon completion of this webinar, what is the expected outcomes? What you are going to gain from this webinar? Upon participation for the next 1.5 hours and listening to my words and speech, what's the gain you are going to get? That's going to be our intended learning outcomes of this presentation. So to explain the strategies and applications of simulation integrated drug discovery with special focus on ligand-based drug design and to acquire knowledge to practice quantitative structure activity relationship modeling simulation for scaling of processes in drug discovery. These are the two achievable learning outcomes I'm going to facilitate you all in today's workshop. However, this is just a knowledge-based component. I'm not going to give any instructional hands-on training, unfortunately. So this is the webinar overview. I will be starting with introduction and I will introduce to you all some simulation strategies in drug discovery, QSR modeling and organization of economic cooperation and development principles for performing a QSR modeling and development. So these are the regulatory practices we generally have and what are the various applications of associated with QSR simulation and in summary references acknowledgements. So moving forward, so let me introduce to you all a brief simple note to the conventional drug discovery pipeline. Why it's so complicated? Because it's a multidisciplinary. So several disciplines actually integrated into a pipeline process, a systematic process. So it's the process of drug discovery. The major problem associated with drug discovery is the time and the expenditure. If you look at the time and expenditure, it's around take 12 years of time and 1 billion plus R&D cost to attain one drug molecule into the market. This is the major challenge. So this is the major challenge associated with the drug discovery pipeline. So as you can see from the slides, there are several stages. Starting from target identification, target assay construction followed by screening and validation, hit to lead optimizations, and so on. And finally, these all comes under the preclinical. And once it enters into the clinical, again, it needs to face phase 1, 2, and 3 three stages of the clinical trials, so that the molecule may enter into the drug market to have it in a clinical practice. So this is the official workflow approved by the Food and Drug Administration, United States. So in total, on an average, it takes not less than 12 years of jury time. So this is the major constraint. So if you look into the whole process, every step is having its own significance, and every step is having its own the desirable outcomes and the regulatory standards, so as to move to the next level. So how can you do progress this all these stages? How to accelerate the process? So is there any way to accelerate the process? Because it's quite very long time, 12 years of duration, and it does matters the money as well. So how is there any other process to accelerate the steps associated with this process? So let's look into what is the major challenge. So the major challenge is day by day, the production cost of the new drug approval process is actually taking a exponential growth. As you can see in the exponential growth of this one, in the 2010s, it's almost reached to 2.6 billions already. So it's quite very high expenditure when in terms of developing a single drug. See, per approved new drug, meaning to say that one single drug. So rather, this is the major challenge. So the drug discovery process associated activities all facing the major challenge is the cost. So is there any replacement or any complementary or any substitution based strategies to complement the process or to accelerate the process in the drug discovery? So let us see. So before we talk about the simulation integration, I just would like to introduce few things about the basic drug discovery process. In the basic drug discovery process, initially the chemical molecule might be exhibited its potency in the form of just a hit molecule. And this molecule, without producing any biological property, this one may be a building block or just a fragment molecule. But once it reaches to the hit molecule stage, meaning to say that it is a bioactive molecule. So this bioactivity, once again, we have to look into the desirable properties of the molecule, whether this molecule can be further carried to next level, so that this molecule can be further modified into the lead molecule. And this lead molecule, whether this molecule is consisting of desirable properties or not, so that they can be further modified into the clinical candidate. So these are all various stages of a single chemical molecule when it experiences throughout the journey of the pipeline, starting from the basic R&D to entering into the market. So actually the basic molecule is a chemical. The basic molecule is a chemical, and that chemical is a fragment. Once it enters into the synthetic process, it becomes the term changes to building block. Once it actually screened through any biological assay, the term changes to, if it is active, the term changes to hit. The hit molecule with good drug-like properties, drug-like properties. So drug-like properties means what? Some desirable properties where the drug molecules mostly have. So such properties, if are there, so it could be a lead molecule. So once the lead molecule exhibiting potency, drug-like properties, as well as the pharmacokinetic parameters, as well as convincing parameters to move forward, so it can be a potential drug candidate, so clinical candidate, so that the preclinical trials can be ended at that point and clinical trials will be started. So a chemical initially as a fragment, and in the synthetic phase, it is a building block, and in the bioassay phase, it is a hit molecule, when it is the optimization process is going on to identify the both molecule, it is a lead. And then when it enters into the clinical trials, it is a clinical candidate. So see the name transformations of a single molecule. But what is the major challenge? The major challenge is the reaching to the starting point to the ending stage. Whether a single molecule could able to reach to that point, so many satisfactory filters need to be crossed. So what are those filters? There are different types of filters: drug-like filters, absorption, distribution, metabolism, excretion parameter filters, and toxicity alerts. Toxicity in the sense, so the molecule, all the types of properties. That process is called prediction. So this prediction is mediated by a simulation. Prediction mediated by a simulation. So what are the various simulation tools and strategies we have? First of all, what is the basic idea of about simulation? So how can we complement these stages in the drug discovery with a simulation process? Is that possible? So let us say, what is a simulation? So what does it mean? According to the British English, there are some definitions. According to American English, there are some definitions. Let us look into the definition. It is just a computational model. So I'm not going to read out the whole paragraph, just it's a computational model integrated with mathematical calculations and supported with algorithms. So we are not the computational professionals. We are not going to develop any algorithms on our own. We are the end users. We are the application chemists or application pharmacists. So we as a application end users, we have to utilize the product. The product is the simulation tool, the computational software or the computational model in a logical way, so as to end up with a hypothesis. So what is the ending here? Hypothesis. So here the process of doing the simulation will only ends with hypothesis. So concept leads to hypothesis. So what else you need to perform? So proof of the concept is the next stage. The proof of the concept study is confirmation to the hypothesis generated by you in the simulation experiment. So an individual who develops a hypothesis must compulsory need to validate the hypothesis. And this process is called proof of the concept process. Without proof of concept, that simulation is not going to consider in any way. It's not going to help the process in any way, because ultimately we have to prove that the simulation hypothesis has been experimentally proven. So the conceptual basis and the simulation experiment has been successfully integrated into the pipeline, because the drug discovery pipeline is not hypothetical process. It is a real experimental process. So what we are planning to do is a computational simulation. Simulation is entirely a hypothetical, an imaginary, an arbitrary pathway. Okay, upon completion of the simulation, we may need to definitely perform the experimental study to confirm the proposed hypothesis. Yes. So what's the landscape of simulation strategies nowadays? So if you carefully look into the landscape, the development of ICT and computational science and the relative fields, they actually booming in three different major areas. So one is artificial intelligence. The other one is machine learning, followed by deep learning. As you can see, artificial intelligence is the ability to imitate intelligent human behavior. Imitate means mimicking. It's just a mimicking the process, but it's not a real process. So this hypothetical process can be performed through artificial intelligence. Robotic hands, the best example is robotic hands. Nowadays, the pipetting, the preparation of the cereal dilutions, all based on the robotic hands. It's the advancement of artificial intelligence and machine learning. When it comes to machine learning, this is the place where the mathematical models will be constructed and developed and validated. So these mathematical models will be more useful to predict information associated with that particular domain. So domain selectivity is more important. So this information we are going to discuss in the next few slides. So domain selectivity, and followed by the deep learning process, application of machine learning, and based on the complex algorithms and deep neural sets as training and test models. So dear participants, I don't want to confuse you all with mentioning so many other background things which will generally happen to develop a algorithm. So according to the chemical basis, we have to construct based on the technology advancements in the background. The algorithms are very important. Algorithms. These algorithms developed based on different strategic principles like Monte Carlo strategies, neuronal networks, and the fundamental basics of this one is conceptualizing the principles and practices of the chemistry and biology with molecular mechanics and quantum mechanics. So these are all basically forms the framework in the background to design a software, and that software can read the chemical language, and that software can produce a three-dimensional model, and the software can predict the information associated with the input database. So here I'm just highlighting here the input. So what is key important strategy? What is the pillar of doing this simulation? What is the pillar? So let us see. So the pillar of doing simulation is the data. If there is no data exists in terms of an experimental activity, so no simulation can be performed, because the pillar of the simulation is the experimental data. So at least one or two experimental data is compulsory. Why? Because if you would like to perform a simulation, simulation is the process of mimicking some process. So if there is no real process exists, what you are going to simulate? So data is the pillar of the simulation. So once the data is, this is the knowledge pyramid. So the data to information, and information to the knowledge, and knowledge to the wisdom. So these are the various steps associated with organizing the information and structuring and developing some conceptual basis for that, and finally interpreting the data. So these are the various steps: context, meaning, and insight. So these steps will be associated with the decision making steps at different levels. So data, information, and knowledge. So the data is the pillar of simulation. Why? Because just assume without an experimental data, with what you are going to perform a simulation, because simulation is the mimicking the real experimental process. So it is a hypothetical process, but you may have to mimic the real experimental process or conditions or steps. So the data associated with not at least one experiment need to be there for the reference to identify the essential steps and to develop a model. So that's what we are going to discuss in the next slide. So why do we need simulations? First of all, why do we need simulations? Because the conventional process is going well, and everything is fine. There are several steps, the regulatory steps, step-by-step process associated with the drug discovery process. But why do we need simulations? Because as I mentioned, the major challenge is the cost, the expenditure. So we need to reduce the expenditure. At the same time, we need to reduce the time associated with the development. So this process must be complemented with some technology advanced tools. So the technology advanced tool which is most related to the drug discovery process is simulation. So these simulations are basically categorized into bioinformatics and chemoinformatics, computer aided drug discovery. bioinformatics and chemoinformatics. How actually this process actually accelerated the process of computational drug discovery. This process actually accelerated because due to the success of Human Genome Project. So because of the success of Human Genome Project, lot of enormous information associated with human genome and structural proteins and functional proteins and genes and plasmids, everything associated the X-ray diffraction structure of the protein is clearly projected, so that we can actually take that experimental information, and this experimental information conceptualize it into information technology mediated by some algorithms, and they are projecting a simulation model for your experiment. So that simulation model can be considered to perform some simulation experiments. So whatever we are trying to do is everything a computational strategy. Just we use the computer, and if you have the sufficient backup and good RAM and supercomputer powering computing power, so that we can accelerate the process. So the only investment required is a computer. That's all. Computer integrated and installed with some molecular modeling and drug discovery softwares. Okay, how this process is going to complement the actual drug discovery pipeline. So in the actual drug discovery pipeline, there is a very big gap. So what is that gap? The gap is associated with the translational gap. Translational gap means from one stage to another stage, when the molecule currently existing in the hit stage. So hit to lead, it is the next stage. So during this translational stage, this molecule could not able to achieve or desirable properties associated with the lead molecule. So this molecule has to be withdrawn from that particular stage. So whatever the efforts, whatever the money, manpower, whatever associated up to that point are wasted, because the molecule is no longer entering into the next level. The molecule has already been withdrawn. So that is the major gap. So how to prevent such gaps and how to try to normalize the curve associated with that gap. So this process can be significantly complemented with computer aided simulations. So computer aided simulations, nowadays, how the simulation actually doing the process in the normal process in the drug discovery, I will show one integrated diagram, how it will complement various stages of preclinical drug discovery stages. So actually this gap is called valley of death. Valley of death. This is the term generally useful. They generally use to address all the gaps in any academic, not only in drug discovery, any industrial setting, any type of concept. If there is a gap that leads to significant amount of loss of economy, so that is a valley of death. So the valley of death is major gap of associated with the drug discovery process. Okay, so building a general simulation model. So how you are going to develop a general simulation model first of all. So as I mentioned before, if you would like to develop a simulation model, you need to have a real system first of all. So a real system, this is the real system, and make a model. And this model has to be integrated with computational algorithms and construct based on the theories, and theoretical predictions are possible. And similarly, the perform simulations using the constructed logical algorithm, and these are the simulation results. Okay, and one way you are getting simulation results, and the other way you are getting the experimental results. So you have the privilege to compare between these two, experimental results and simulation results. In what way you can make use of this comparison? This comparison can clearly shows what is the percentage degree of deviation, the root mean square deviation associated with the predicted and experimental results. In case if the correlation coefficient, the predicted error sum of squares, and the F statistic, and so on. So several statistical measures we can make use of it to identify whether the correlation is actually good or moderate or less. So based on that, we can consider this model is reliable. So for every single model, there are several parameters to satisfy. These parameters are statistical parameters, and these parameters are well defined to individual type of technique we are using. For example, if you are using multivariate statistics like multiple linear regression, principal component analysis, partial least square technique, and so on. So compulsory, there are several statistics. The statistics, cross-validation statistics like leave one out method, leave five out method, and cross-validation correlation coefficient of the training set and test set. So there are several statistical measures. So I'm not going to talk about the individual statistics because that is experimental protocol. What I would like to highlight here is whether there is any correlation between experimental and theoretical predictions, the data. So the data obtained from the experimental conditions and data obtained using the simulation conditions, are they having any close correlation? If there is any correlation, the model is considered as validated, and that model can be further subjected for screening process. So based on that model, a new component can be introduced into the model and identify the possible orientation of that model within the simulation. So the model can project the predictive power of that model, simulation experiment. So what is the predictive power here? Predictive power means whether this can be subjected for the next level or not. So such decision making information can be provided. So decision making information can be getting from the simulation models, whether they are reliable or not. It totally depends on several factors. There are several factors, n number of factors, and this process is called curation. This process is called curation. So if your data points are curated, well curated, so definitely your experimental data that can produce the predictive data that produce based on the simulations surely will have some correlation. In case if the data is non-reliable or non-curated, so there will be a lot of deviation from the expected values. So this is how generally it will take place. So basically, if you would like to develop a simulation model, it's not only to the context of drug discovery, any kind of experiment, you need to have a real system. So based on the real system, based on the availability of the domains, based on the availability of the system algorithms, we can construct a simulation platform, and that simulation platform will be used as a virtual screening tool. So the term virtual screening tool. So what is virtual screening? Whatever you perform using computer is called virtual screening. Okay, screening a set of molecules using a computer. So I'm going to introduce few strategies so that you will have better understanding on this. So how you are going to, so just now we discussed about the basis of some simulations. So how we are going to integrate into different steps. So these are the basic steps associated with the pipeline of drug discovery. I would like to highlight one more thing here, dear participants. None of the steps in the actual pipeline will be substituted. Okay, meaning to say that every step associated with the actual pipeline process will retain as it is. There is no change at all. The only change is the amount of information you are going to subject inside. So the amount of the number of in terms of screening, the number of compounds you are subjecting for the screening. For example, in the target identification, if you compare the conventional way, there is no complement of the bioinformatics or docking studies or chemical biology. What we have to do? There is no alternative. We may need to screen whatever the possible biological macromolecule is present in the database. To till date, every single molecule must be screened because there is no complement information. There is no rationale. Where is the rationale? So not possible. So we need to progress through across all the database of real experimental proteins or macromolecules. But in case of this one, bioinformatics, reverse docking, and computational biology, they provide some outcome. And based on this hypothesis, they can actually screen a lot of information and they can narrow down the information which they would like to subject for this particular stage. And similarly, moving forward, the target druggability and the computational systems biology, because since last 3 decades, the progression and acceleration of the computational simulations is very well recorded in the drug discovery history. Because as we all know, majority of the drugs, anti-HIV compounds are entered into the market based on the structure-based drug discovery strategies. And quinoline antibiotics are specially approved based on the ligand-based drug discovery strategies. So there are good number of success stories we have based on the simulations. We have some success stories. So based on those success stories, we are still practicing the simulation-based screening and complementing the actual pipeline of drug discovery. So this is totally a complementary process, but not at all a replacement process. Meaning to say that none of the steps associated with the actual pipeline will be replaced. So every component, every stage will retain as it is. So any additional hypothesis you generated sideways basing on the computational software, so that can be directly entered into that particular stage and try to see your luck. That's all. It's the proof of concept. If concept is proven, you are lucky. If concept is not proven, at least you managed to save some money and you managed to save some time and the manpower associated. That's all. That's all about the simulation integration. Otherwise, why we need a simulation? Because the real experiment anyway we have to do. Why we want simulation? There is no alternative to, there is no replacement for the real experiment. So simulation can reduce the number of experiments. So dear participants, simulation can reduce the total number of experiments. If you perform, the total number of experiments are 3000 in case of high throughput screening. But if you mediated with this computer-aided drug discovery, the 3000 experiments may be narrowed down to 200 experiments. So nearly 2800 experiments you have compromised. Why? Because you have some rationale. And this rationale generated based on validated, conceptualized computer-aided drug discovery practices and software applications. Okay, let us move forward to look into application of AI in drug discovery pipeline. In what way artificial intelligence useful in the drug discovery optimization? There is a small analogy here, lock and key type of analogy. So if you can consider here, finding locks for new doors. So finding locks for new doors, whatever we are talking about from this point of slide, so everything is associated with computer-aided drug discovery. So the simulation-based activity. So I have to highlight here one thing, the scaling up processes. But in the drug discovery pipeline, there will be scaling up and also scaling down processes as well. Because scaling up processes in one aspect, it is very important. Once upon completion of all the spaces of drug discovery and marketed drug is entered into, then this process is very significant. But once before it enters into the clinical phase, the scaling down process is very important. Why? Because scaling down process is the cost effective. whereas scaling up process, that means generating a lot of chemicals and the intermediates and active pharmaceutical ingredients, new chemical entities. So this is all cost effective. No, not cost effective. This is all a lot of expenditure. So keeping that point in view, scale up process and scale down process will be interchanged in different levels of drug discovery. So here, one thing I would like to highlight, when the scale up process is important, well, the scale down process is important. Actually, the scale up is also associated with not only the experimental process, it also associated with the simulations. So what I would like to highlight here is scale up simulations. Scale up simulations. Scale up simulations means scale up simulations will scale down the experiments. Scale up simulations will scale down the experiments, so that the simulation models, the reliable simulation models will be reposited in the database and readily available to screen the billions of molecules, the chemical diversity, to identify the suitable bioactive molecule, the proposed virtual hit molecule. Virtual hit molecule. When this virtual hit molecule is going to be the real hit molecule? Upon successful performance or conduction of the experimental study. So then after performing the experimental study, you can see that very clearly whether the compound is exhibiting any bioactivity or not. If a compound is exhibiting some bioactivity, consider that your concept is proven. So proof of the concept study is successful, as we discussed a few minutes back. So if these are the various integrated simulations possible at different stages. So this slide is a general overview. So I'm going to discuss in detail each strategy. So simulation integrated scaling processes, because the scaling processes sometimes scale up process is needed and sometimes scale down process is needed. Okay, let me give an example. When it enters into the lead optimized state, meaning to say that the lead molecule is already a potential molecule that can enter into the clinical studies. So at that point, if the potency of the molecule is close to 10 micromolar concentration range, but the micromolar concentration range, you would like to have nanomolar concentration range, what you can perform? At that point, you will be performing the scale up synthesis. Scale up synthesis. What are the strategies? Combinatorial study. The combinatorial parallel synthesis, solid phase synthesis, strategic process, we can make use of it at that point to identify different derivatives, to study n number of molecules associated with that lead molecule. And in case if any molecule exhibits potential biological activity at nanomolar concentration, so your scale up process is successful and move forward with the clinical phases. Okay, so like that, the simulation experiments will also enable a researcher to decide whether to perform the scale up process or scale down process associated with different strategies. So this is an integrated protocol where you can see both the experimental protocol is also present and the computational protocol is also present. This is the experimental protocol where the known actives, compulsory, the known actives must be there. So, and this is the protocol where the X-ray diffraction derived this functional protein. So in case, for example, the functional protein, the three-dimensional protein, the tertiary structure of protein molecule is not available in the database. In such case, what we can perform? Only the sequence managed to isolate, but not a target protein molecule. In such case, we may follow some computational strategy, that is homology modeling. Okay, but one of the best strategy that can identify the three-dimensional orientation of the biological macromolecule is X-ray diffraction study. So the X-ray diffraction is one of the best technique to elucidate the complete structure, the three-dimensional Cartesian coordinates of the biological macromolecule. And that information is going to be the basis for developing the simulation. in a molecular docking study, homology modeling study, fragment-based drug discovery, and so on. So based on experiment, we actually developed some simulations. So compulsory and experimental evidence is the bridge to develop a simulation based model. Okay, so moving forward, let us see. Just this is some illustrations, graphical animations to show how the simulation generally works. So protein interaction and protein DNA interaction. So as you know, the COVID virus is actually interacting with our angiotensin converting engine as one of the key major targeting protein. So what type of interaction could be happening there? So you may do some simulation experiments on that by considering, because the complete COVID main protease is already clearly resolved. The NMR experimental resolution is clearly reposited. So we have the evidence of the three-dimensional structure of the main protease of the COVID, and you have the evidence of the angiotensin enzyme, angiotensin converting enzyme of human, the real structure. So you can perform some simulation with the protein docking study. So for that, you may use some software tool, which is most widely used one. Hex is the tool they generally make use of it. Do the protein docking and protein DNA docking and so on. So, and similarly, DNA ligand interaction, where you can see this DNA ligand interaction as we study in the anti-cancer agents, alkylating agents, nitrogen mustards. These are the compounds which can alkylate the DNA base pairs. So this is the process. So this is the simulation that can enable the computational chemist to identify the specificity of the molecule at that particular target site. So once the target site specificity determined or predicted, so confidently may go for proof of concept study. If this information is not supported, so we may be performing a blind screening without any rationale. So that is the iterative process. It's very tedious. So definitely nowadays the acceleration is mediated by the simulation. So the other one is protein ligand interactions. So sometimes multiple conformational docking study is one of the advanced one. Why this one? Because the receptor conformational states, the induced fit, conformational perturbation and non-conformational perturbation, flexible docking, rigid docking, like this different types of simulation models are available. So which model you would like to utilize for your identification of some novel molecule to test in the real experiment, that model you are happy to select, and you can proceed with the experimental design. So moving on, this is the homology modeling, as I mentioned, protein with unknown three-dimensional structure, especially. What that means, the isolation is only possible with only the amino acid sequence, but without having any three-dimensional structure, because the X-ray diffraction, the crystal may be not stable and could not withstand the pressure in the cell of the X-ray diffraction unit. So that may be the reason. So in such case, what we can do? We can consider the template target protein, because due to the advancements of the algorithm, this type of simulation we can perform. The simulation is called sequence alignment, and upon performing the sequence alignment, the target template sequence alignment is possible. So the target means the sequence where three-dimensional structure is unknown, and the template is actually the three-dimensional structure is known, and the sequence of the three-dimensional structure having some close structural resemblance, the sequence similarity with the query sequence. If the query sequence is having some similarity, and that process can be mediated, not less than 30% similarity should be there, meaning that at least 30% similarity is the minimum standard to perform this strategy. So upon developing the model, there are several what we say stability analysis, like protein side chain flexibility, stability analysis. Of course, the better, the most reliable tool to analyze the stability of a protein developed using homology modeling is better Ramachandran plot statistics. So the Ramachandran plot statistics is will clearly project the there are several zones, that means allowed, disallowed regions, and additionally allowed regions, generously allowed region, and most favorable region. So these are the various zones associated. You can see the phi and psi angles, the dihedral angles of every amino acid with restricted rotation will have to fall in that particular zone. So in case if the amino acid is falling in the disallowed region, meaning to say that the actual target protein sequence has been disrupted or damaged or cut into fragments. Something might happen. It's not a good quality sequence because the sequence has damaged, because the clear indication of the experimental observations, because the G.N. Ramachandran was the actual developer of this concept and a Nobel laureate as well. So in European Union, RCSB protein data bank, everywhere the Ramachandran plot statistics is the key standard for the protein validation. So keeping that point in view, we may validate our homology modeling, whether we can do some simulations associated with that model or not. Okay. So moving forward, the next one, if the protein structure is available, and binding site information is partially known, meaning to say that partially known. How do we say partially? How do you say fully known? It depends on the value and its binding affinity. So what is the binding affinity value of that particular ligand and where it is going and binding? So based on that, we can determine whether it is a partial or fully binded ligand or something. So the incremental construction is the strategy associated with this simulation, incremental strategy. So these are all the building blocks. So the fragments. So these fragments will be joined together to construct a molecule. And this construction may exhibit some binding properties within the binding site region. And this binding site region may be varied. This binding properties may be varied from compound to compound. So this process of variation may take place, and how it directly reflects the binding biological activity as well. For example, if you could consider here, the typical fragment is just a IC50 value is 1 millimolar concentration. So 1 millimolar concentration. Upon incremental construction, if you could consider the occupancy of the receptor binding site is well occupied and the fragment has achieved 10 nanomolar concentration, meaning to say that the biological property has been enhanced due to the occupancy of the receptor and due to the binding potency of the ligand at that particular receptor. So this is all about the fragment based drug discovery. So this fragment based drug discovery basically constructed once if you know the three-dimensional structure of the protein and any experimental ligand but not a drug molecule. Okay, in such case the by keeping that loophole as a tailing point, and we will proceed further to identify the big constructed molecule to establish a proposed binding affinity. So this is also a rational drug discovery process as well. Moving forward, the molecular docking simulation, as you can see, mushroom growth of publications nowadays with respect to molecular docking simulation. Every single graduate, postgraduate will do some docking simulation and screen the database, and they will say these are the potential binders. These are the potential binders. So if you look at such article, generally, I'm not just criticizing, just I would like to explore further because it's an explorative research. This is not a conclusive, it's an exploratory research. So as a matter of fact, if you consider such type of hypothesis, what is the evidence? So there is no proof of concept, Okay, once after you get some evidence, so that's just a virtual, hypothetical, imaginary, or it's just a narrative conclusion, but not it is a reliable or carry forward thing. And in many cases, you can see the solvation properties are ignored, and the water molecules, especially inclusion of water molecules, if you perform the docking studies, what is the variation, and the exclusion of water molecules if you perform the docking study, what is the variation? Because in the real experimental setting, you will, in the real biological environment, you will have biological fluids, cofactors, enzymes, and so many other complex molecules. So within the complex molecules, what is the actual binding? So the molecular dynamic simulations are nowadays gaining a lot of popularity. MDS, molecular dynamic simulations. Yeah. So the molecular docking is also one of the strategy, well-known simulation, when this simulation can generally be performed. If the clinically used drug molecule having co-crystallized with the protein target, and that is the very good evidence to perform molecular docking study. So dear participants, what I would like to say is, if you would like to perform molecular docking study for any specific drug target, please make sure that the drug target consisting of some experimentally proven, clinically used drug. Because since the clinically used drug molecule, which is already proven to be effective in terms of pharmacodynamic and pharmacokinetic parameters, and not only that, the ligand specificity at that binding domain is very selective. Okay, so those properties are significant information. So such significant information will make your hypothesis more strong and your research question more reasonable. So keeping that point in view, please try to consider the standard. It is all about the standard of simulation you would like to perform. If your standard of simulation is just a blind screening, yes, of course you can do that. But will you be carrying forward to the experimental study, or you are going to stop there? So what's your decision? So your decision is also will be confident based on your selection of the protocol. Okay, so protocol selection and putting the standards at every stage, and the curation is very important. And we are going to talk about the curation with specific role of QSAR modeling. So now the next one, so ligand based drug design simulations. So ligand based drug design simulations, in case, for example, there is no specific information associated with the protein target, only ligand molecule is known, no other information is known. In such case, what we are going to do? But still the molecule is approved as a potential drug. So I just would like to ask, give me one example where the specific drug target is unknown. I can say paracetamol. Even paracetamol, there is no specific and selective mechanism of action on the target side is elucidated till date. So meaning what? It is just a ligand showing a potential antipyretic, anti-analgesic activity, So you may ask me, then why we are still using? Because the compound is safe and efficacious. Since the compound is safe and efficacious, and we have the therapeutic benefit of using paracetamol and relieving the symptoms of fever and relieving and also the relieving the symptoms of the body pains and so on. So we are happily using that molecule and enjoying the therapeutic benefit of associated with that molecule, irrespective of whether where it is actually binding. So that is the safety and efficacy profile of that molecule. So there has been one more strategy developed in such hypothesis, that is ligand based drug discovery. So the ligand-based drug discovery simulations associated with only the drug molecule without taking into consideration of any drug target or binding interactions or any other profile, only the molecule. For example, the only drug molecule which is currently existing for the treatment of COVID approved by FDA is the only molecule is Remdesivir. So based on Remdesivir, so many fragment-based and metabolomic studies are going on. They are developing different combinatorial strategies because Remdesivir is only the molecule currently approved for the treatment of COVID by US FDA. So keeping that point in view, Remdesivir is considered as a ligand, the target ligand. Keeping that ligand into the query, the database search, the virtual screening process will be performed, and we can identify some related analogs to synthesize in the laboratory and further testing using the COVID cell lines or animal models induced with COVID and so on. So this is how we can move forward. The simulation is going to complement the process, and simulation is going to support your imagination. The simulation is going to complement the time, expenditure, manpower associated with the drug discovery. So let us see one by one. So the first strategy, the basic strategy associated with it, simulation search. What is the rationale of this simulation search? If the molecules both are similar, the biological activity also will be similar. So that's the basic rationale of this. So this can be computed based on the database, a query molecule. For example, just imagine this molecule as the Remdesivir, and this is the database of molecules. So Remdesivir virtually screened against database. So what is the score? The numerical representation of the similarity will be the score. The numerical representation of the similarity will be the score. For example, the top score will be the 1. 1 out of 1 is the 100% similar molecule. 0.9 out of 1, 90% similar molecule, and so on. So you can collect the top 5, since it is just a simulation experiment. You can collect top 5, 1, 2, 3, 4, 5, and move forward to the experimental study and try to identify whether your concept is proved well or not. So the proof of concept study initiated. So if the proof of concept is successful, you can happily move forward to the lead of hit optimization and lead identification and so on. That is how the similarity search simulation complementing the process of the drug discovery in scaling up the compounds. So next one is the advanced strategy associated with the three-dimensional properties, steric and electrostatic, that is pharmacophoric compound. So pharmacophore modeling, and this pharmacophore model simulation basically consists of the chemical features, the whole molecule, because this one in this case this is an atomic similarity. That means each and every atom will be taken into consideration. Every bond, every single atom, hybridization, everything will be taken into consideration with respect to this one. However, in case of pharmacophore modeling, only the key features means hydrogen bond acceptors, hydrogen bond donors, halogen moiety, ring system. The ring system can be a benzene ring system or pyridine ring system and so on. Different types of ring systems. So the categorization is based on the types of chemicals, the types of chemical features. Types of chemical features: hydrogen bond acceptor, positive charge, negative charge, hydrogen bond donor, ring system, and hydrophobic atom. So these are the basic chemical features will be considered. So what are the parameters they generally simulate to prioritize the molecules? What are the parameters generally they will use to prioritize the parameters? So the parameters are, one is bond angle and the other one is bond distance, interatomic bond angle and interatomic bond distance. So these two are the parameters we generally consider to construct a pharmacophore model. Based on this model, irrespective of whether the congeneric series or non-congeneric series. So I have to explain very good differences between similarity and pharmacophore. When if you perform the similarity search, most of the times you may get the congeneric series of molecules, meaning to say that the molecule belongs to the same background basic skeleton. Okay. But if you perform a search using the pharmacophore model, you will get different types of variety of chemicals. So these different types of variety of chemicals, meaning to say that the diversity of the chemical space has been enhanced. So the diversity of the chemical space has been enhanced. You will be getting a lot of opportunity to screen different orientations of different types of molecules rather than looking at the same chemotype with the different side chains. So this is one of the advanced feature of the pharmacophore modeling. However, the pharmacophore modeling has been further integrated into some other technique as well. So that is actually integrated simulations. That means also we say hybrid simulations. Okay, let me explain to you, dear participants, what do you mean by hybrid simulations? So as a simulator, as a computational simulation scientist, you developing a protocol to identify a hit molecule or to prioritize a hit molecule. Okay, so what are the strategies you integrated? So the first strategy I would like to consider is ligand based drug design similarity strategy. Okay, first strategy is similarity. So the in the protocol your first strategy is similarity. Okay, next, after similarity you identify the top 5 molecules. Okay, these top 5 molecules, if in case the established structure of the protein is already available, plan some docking studies for these 5 molecules. Okay, so after doing the docking study of these 5 molecules, you will get top 1 hit molecule. Okay, so that top hit molecule, develop a new strategy, that is pharmacophore model for the top hit molecule, and do the screening again. So you will get a diverse range of chemical moiety, and because of the similarity, the molecule is top molecules will be considered as docking studies for the binding properties, and followed by the pharmacophore identification. And so you have developed an integrated model for the screening. So there is no specific logical way of doing computational simulations. There is no established strategies for this. You as a researcher, you can design your own strategy. As a researcher, you have all liberty to formulate your own simulation workflow, because ultimately what is more important is whether are you going to prove the concept with the experimental study or not. As long as you have the complete confidence to prove it in the experimental proof of concept study, you can design any kind of simulation into your protocol. You can integrate different strategies, and you will end up with one molecule. Certainly you did the molecule screening in the in vitro experimental conditions, and you have identified that molecule is potential bioactive molecule. That's all. Your protocol is highly validated, and it can be useful as a screening tool and benchmarking tool for the upcoming compounds as well. So that's how we can move progress and the scaling of the simulations and integrated processes in the drug discovery. Okay, now we will be entering into a specific strategy that is QSAR. Why I would like to highlight here QSAR more deeply. Why? Because as I mentioned just now, anything you can put inside and formulate your own strategy. Yes, you can do that, provided the other all the strategies does not have a standard regulatory authority to say that okay, this is the standard and this is not standard. But in case of QSAR, the simulations what you can perform must meet certain principles developed by regulatory bodies. And that's the reason why QSAR is the only regulatory tool used in the simulation experiments. Is the only regulatory tool, meaning to say that it is a standard tool in a regular practice. However, the other strategies also can be used as a regular basis, but they don't have any specific supervision or regulatory authorities to say that this is true and this is correct, and because the diversity. If you look at the diversity, lot of constraints, so many constraints, especially the major constraints associated with all the receptor based one is solvation properties and crystallization behavior, polymorphism. So different problems are associated. But in case of QSAR, it is totally a mathematical model, a statistically validated model, and the clear distinction and discrimination can be made between the isomers or the meta and para. So such discrimination will clearly project the numerical integration of the relevant activity. So what is the dependent variable and what is the independent variable here? So if you are performing a quantitative structure activity relationship, always the activity will be the dependent variable. Whereas the molecular descriptor is nothing but a property, a physicochemical property or topological property, any property. So a property that is a independent variable. So in the I would like to acknowledge these two great innovators of the QSAR methodology. 1874, Wilhelm Korner clearly distinguished the substituted differences of benzene, ortho, meta, and para, and 1964 clearly identified there is a direct relationship exhibited from the lipophilicity and the alveolar concentration. And other things, and we can also identify there is one theory stated based on this, that is Mayer-Overton theory. The Mayer-Overton theory is also deals with there is a direct correlation between the lipophilicity and the alveolar concentration for the general anesthetics. So like that, there are several strategies actually developed, and this is the well-established mathematical model and relationships. These are the two contributors we can acknowledge. And moving forward, and what is the molecular descriptor? As I mentioned, molecular descriptor is not an experimentally derived component, but the molecular descriptor is the final result of a logical and mathematical procedure with the transforms chemical information encoded with the symbolic representation of a molecule into useful number. For example, useful number means, so molecular weight 500 and molecular 400. So this number clearly indicating the number of carbons, hydrogens, nitrogens, and their atomic mass, everything. So similarly, it must be a useful number. So that number must have some interpretation with the chemical structure. So if the number does not have any interpretation with the chemical structure, no use. That's not considered as a molecular descriptor. Why? Because the molecular descriptor always gives some chemical structural information or chemical information of a molecule, and it must be a dependent variable in a such a way that change in the there must be a direct proportionality. That means the change in the number, change in the chemical structure must reflect in the change in number, and change in number also reflect to the change in chemical structure. So that direct proportionality should be there. So then it could be a good molecular descriptor. So there are n number of theoretical molecular descriptors developed in recent days to identify a reliable mathematical model to screen the compounds based on the properties. Okay, so but here, dear participants, one thing I would like to highlight here, in terms of quantitative structure activity relationship, there is a small constraint. There is a small constraint. What's that small constraint? Is that you need to have statistically significant number of molecules. Statistically significant number of molecules to construct a model, and statistically significant number of molecules to validate the model. So model construction, model building, and model validation totally depends on the data set. So if you don't have the access to good number of compounds data set, QSAR is not the reliable strategy, because QSAR totally depends on the statistical parameters. So data set is the key component of the QSAR model, data set. So if you ask me what is the statistical number, I would say statistically significant, the training set n is equal to not less than 20. Yes, n is equal to not less than 20. That means 20 molecules with predefined biological activity data. 20 molecules with predefined biological activity data means which activity? Okay, you may be looking forward to sedative and hypnotic, and I may be interested in chemotherapeutic, anti-cancer, or some other person may be interested with some other activity. It doesn't matter. So the thing is, you need to have a significant number of compounds and a desired biological activity, and that biological activity also we have to be curate the molecules and biological activity parameters very carefully. So the curation process is a strategic process. And before that, let us see what are the ideal properties of a molecular descriptor. First of all, which parameter we can consider as a molecular descriptor, which one cannot. So this is the one of the best reference to look into molecular descriptors for the chemoinformatics, and should have structural interpretation. As I mentioned, the structural interpretation. So if there is no relationship between the descriptor and the chemical structure, that's not useful. And you have good correlation with at least one property. So good correlation with at least one property in the sense, because as you know, quantitative structure activity relationship studies can be performed not only to the biological activity point of view, but also to the property calculation as well in the pharmaceutical formulations especially. And that strategy is called QSPR, quantitative structure property relationship. Okay, and the other strategy is there that is very, very important, especially to identify the molecules which are very poisonous or consisting of very toxin something. So what is that strategy? QSTR, quantitative structure toxicity relationship. So actually the background principles, strategy, everything is same. Instead of biological activity, the property means, for example, Weiner's index, Andrews affinity, solubility coefficient, distribution coefficient, and so on. So there must be property dependent relationship. So for that reason, we have at least one property good correlation must have. And similarly, preferably discriminate among isomers. Discriminate among isomers means, because since isomers are close to chemical structures, resembles the same chemical structures, so there must be some discrimination. If there is no discrimination, how can you identify which isomer is that? So for example, the best thing is optical rotation. So optical rotation can see very clearly the chiral isomers in different orientations, and should change gradually with the change in structure. As I mentioned, in case the number changes, chemical structure also must change, and in case such structure changes, the number also must change. So such type of direct relationship. And you have the correct size dependence, it related to molecular size. So correct size dependence in the sense, this is actually somewhat ambiguous. For example, if you get very big number or 4 or 5 digits number, but very minor change in the chemical structure related to one atom or two atom, that significantly gives a lot of precision, and that would be what we say generally. It is a it leads to the pain type of process. The pain type of process means it is one of the strategy where we will have trouble. We will have trouble with that kind of compound, because the process is what will happen when you are constructing a linear curve, that is overfitting will take place. Overfitting. And due to this overfitting, because there is a very narrow change that really does not reflect on the chemical structure a lot, so it may lead to some process change as well, and should not be based on experimental property. Why? Because if you are performing experimental property once again, everything, what is the objective of doing simulation? Because the main objective of doing simulation is to reduce the total number of experiments. So it should not be based on an experiment. It should be based on some theoretical background. That means the different, what we say, the basic one, very potential, yes, topological parameters. Topological, the three-dimensional topological parameters we can consider, because they are not actually based on the experiment. They are based on the electron cloud density and steric and electrostatic repulsions of the compounds. So theoretically derived descriptors are more fruitful to develop a model rather than an experimental property. Okay, moving on. Next one, data set curation. I may be spending some time in this slide because this is one of the key component. You are going to learn most significant information from this. If I am not mistaken, how much time I have still? May I know how much time I have still?
Another 15 minutes.
Oh, we have another 15 minutes. Okay. I hope I will try to conclude before that. So, dear participants, this data set curation is very significant step before we start doing the simulation. Because if the data set is like raw material, if you would like to develop a product, your raw material must be quality raw material. So data set curation is totally a raw material. So how to make it more quality raw material? So if you apply these filters, the raw material, that is the data set, could be screened with all the types of errors and all the types of ambiguous elements in the data, and the data will be normalized and equal data, and it will be very good data. So let us see what are the various types of filters we can apply. So removal of mixtures and inorganics. So in the first step, removal of mixtures and inorganics, and structural conversion, cleaning, removal of salts, and normalization of specific chemotypes. Why normalization of specific chemotypes? Because in this case we have to focus one thing. QSAR is mostly for congeneric series of compounds, but not for the non-congeneric series of compounds. Meaning to say that the series must be belongs to same chemotype. The basic nucleus must be the same one. If the basic nucleus is barbiturate and the other one is benzodiazepine, if you want to keep it in a same training set, that's wrong. Because the normalization is variant here, because this is a barbituric acid chemotype, that is a benzodiazepine chemotype. So that normalization must be different. And any duplicates in the sense, for example, two molecules may sometimes produce the similar activity. In such case, what is the distinct discrimination we can produce? Here it's very less. So duplication removal is very important. Next, manual inspection. Certainly after doing all the things, we have to also check the manually, so that next step we can proceed. Duplicate analysis and intra lab variability. So intra and external lab variability, this is more towards the biological experiment, because one time if you do the screening, oh, very potential compound, nanomolar concentration, the moment you repeat the experiment, none of the results are reproducible. Meaning to say that there is no intra and interlab experimental validity. Okay, so the exclusion of unreliable data sources, meaning to say that, so which type of screening bioassay you are used? Is that bioassay recommended by US FDA? Is that bioassay the activity in that bioassay can be considered as the benchmarking standard to go for the next process in the drug discovery? So such type of things you need to cross check the reliable sources or not. And detection of verification activity cliffs. As I mentioned, activity cliff means the closest values, and change in chemical structure also, there is no marginal change in the biological activity, that is activity cliff. That means it already saturated at one point, so no major changes will happen. So calculation and tuning of data set modality index. So this one we have to calculate whether the significant number of molecules are present in the medium, low and high. All categories of compounds must be part of your data set. If your data set is totally a high bioactive compounds, so that is actually will not give you the predictable model, because you are expecting the prediction falls within the range of high activity, moderate activity and low activity. So the modalities must cover the range from least active, moderate and high, so that the model developed by using the simulation can be more predictive power, that can have the more predictive power. Yeah. So consensus QSR prediction to curate mislabeled compounds, and this one mislabeled compounds in the sense this is all about removing some duplicates or stereogenic isomers and so on. So certainly you will get from original set to curated set, original set to curated set. Okay, so this is the process, very significant process, where chemical and biological curation. So the chemical biological curation, after that this data is eligible to develop a model. So the curated data set is only eligible to develop a model. So the original data set as it is, we cannot actually consider for modeling because of these limitations. So upon performing the curation step, we can consider carrying forward. And moving forward, this is the strategy. As you can see, the starting input is the step-by-step indications here. Curated chemical data set, descriptor generation. As I mentioned, descriptors may be physicochemical properties, topological properties, any kind of properties as well. And splitting into two sets, training and test. Training is the model development set. Test set is the model validation set. Okay, so model development, validation, and once validation successful, if the model validated and screened all the database of compounds, the model validated and successfully fulfilled all the parameters associated with the statistics, then that model can be used as a virtual screening tool, a simulation tool. So virtual design and further prediction is possible, and proof of concept with experimental studies, as we know. So biological response is equal to F into molecular descriptor. Here F is the mathematical function, a statistical operator. So different types of mathematical functions can be integrated into QSR modeling. There are n number of techniques. So that one we can have the hand when you are doing any hands-on experimentation with the QSR building and validation, you will come across with the software protocols. So now I would like to introduce to you the OECD principles, Organization for Economic and Cooperation Development principles on QSR. This is the regulatory requirement. So just this regulatory requirement, it is the additional information for you. Why? Because if this information is available, the documented for your model, then your model can be adaptable into regulatory process. If this documentation is not fulfilled, any of the items are missing, none of the principles are fulfilled by your model, that model is not useful for the regulatory purpose. So these are the, in case if you are ambitious to make your model available for the regulatory processes and as a screening tool for the upcoming generation of the compounds and chemical diversity, these principles need to be taken into serious consideration. And principle one, a defined endpoint. So what is the actual endpoint of the bioassay? So very clearly defined. An unambiguous algorithm. Unambiguous algorithm means the step-by-step, the logical steps interconnected to each other for the working of the whole software must be clearly shown. Meaning to say that what is the integrated background language? Is it Python or BioPython or C++ or what type of logical algorithm you actually used? So there must be clear indication that how the parameterization is going on and how the model is actually building the computational science. Okay, defined domain of applicability. What is domain of applicability? As I mentioned just now, Domain of applicability means this model is specific to what? This model is specific to screening of anti-diabetic compounds with 10 micromolar to 30 micromolar range. So 10 micromolar to 30 micromolar range. And what is the chemical type? Sulfonyl ureas. So that is the domain of applicability. So sulfonyl ureas can be screened using this model, and the sulfonyl ureas range could be 10 to 30 micromolar. So whether the compound can be identified or not, that is the domain of applicability. Moving forward, the next principle is appropriate measurements of goodness of fit. As I mentioned, the goodness of fit is same as we see in the correlation statistics, the linearity, and the robustness and predictivity. So this one is robustness based on leave one out method and leave five out method and leave three out method. This type of things we can do. And also sampling, what we say, the different types of sampling processes also can be inculcated, meaning to say that you can take out some data set from that and you can check the predictivity and reliability of the model, and you can take out some values, and you can further check for the possibility to prediction. So all these types of different types of strategies are there to test and to validate. So next one, a mechanistic interpretation if possible. So that mechanistic interpretation meaning to say that mechanism of action. So mechanism of action, how these things are actually happening, and what is the in vivo mechanism of action associated. This one is an optional. If possible, we can provide the documentation. If not possible, it's okay. But principle 1, 2, 3, 4 are significantly very important. Okay, so this is some additional information. I provided some text. I'm not going to read out. And the same thing what we discussed is available in the text form. You can make use of this information when I provide the slides to the organizers. So this is some additional information only. So now at last,
Excuse me, sir. We have another 2 minutes to close.
Yes, we will be completing now. So the major errors that affect the OECD principles in QSR development. These are the associated, the principal variations are associated with reliability and reproducibility. Okay, reliability and reproducibility are the critical issues in simulation experiments. This is one of the example of a 3D QSR model as we developed in our IMU. And applications, when you consider the application point of view, every stage of the drug discovery, QSR is having its own role. That is the reason why QSR has been considered as one of the regulatory tool to use the virtual screening process in ADMET, hit prioritization, optimization, and ligand affinity interactions everywhere. So lead optimization and lead identification also identified as most important steps integrated to QSR simulation. These are some successful outcomes of QSR. In summary, we would say the real breakthrough in simulation science will come when we use the simulations as one of the reliable tools to ensure that only active designs are useful for scaling the processes. So meaning to say that when we get the opportunity to completely replace some number of experiments through simulations and reduce the total number of experiments to a single digit experiment, that is where the really simulation experiments are useful to scaling up the processes in the drug discovery. But as of now, we have not reached to that extent based on the evidences. So the take home message is keep calm and use a simulation. That's all we can do. So these are some related references used to construct the slides. My special acknowledgements to distinguished members, starting with the Associate Professor Muhammad Julkifli, Dean School of Pharmacy, IMU Malaysia, followed by Dr. Gansuke, Associate Dean, Academic School of Pharmacy, IMU, and Professor Mallikarjuna Rao Pichika, Associate Dean, Research, School of Pharmacy, IMU, Dr. Lee Choi Sin, Head of the Department, and Professor Dr. Y. Rajendra Prasad, my mentor and project supervisor, Andhra University College of Pharmaceutical Sciences Principal, and Professor Dr. T.V. Narayana, National President, Indian Pharmaceutical Association, India, Professor Dr. S. Vijayadhara, Chairman, IPA Education Division, India, and Dr. R. Srinivas, President, Dr. Sri N. Srinivas, Secretary and Correspondent, CHIPS India, and Colonel B. Venkat, Director, Faculty Development, AICTE, New Delhi, Professor Vijay Vaidya, Executive Secretary, ISTE, New Delhi, Associate Professor Dr. Venkatrao Utla, CHIPS India, organizing members and beloved participants, press and media. Thank you all for the opportunity. Thank you once again. And in case if you have any questions, I am here to interact with you all. Thank you.
Thank you, sir, for the detailed presentation. So I request participants can drop their queries in the chat box so I can put forward to the speaker. So before that, I have a question.
Yes, please.
So when we have training in these experimental models, there will be chances of less errors. But when we use this type of computational techniques, what will be the accuracy of it?
Yeah, that's what I addressed in many instances during my presentation. That is, the proof of concept study is compulsory. What we are talking about is the before proof of concept study, the conventional strategy. The conventional strategy is it is there in its place. There is no strategic convention for that. There is no limitation for that. We can happily proceed further. However, and then what is the advantage of getting new tools or new strategies or technology advancements into the process? So day by day, we are trying to integrate several new things, moving forward with the technology and trying to integrate such tools into our regular conventional process and trying to complement the things. Yeah, that's what we are trying to do. Yeah, if you ask that, of course, definitely I would say experiment is the highest form of the research.
So are there any free software for using this molecular simulation modeling techniques?
Yeah, good number of softwares we have. We have good number of softwares. The only problem is, as you mentioned, reliability. The reproducibility of the information is very poor. For example, if you run one simulation experiment, if you run the same simulation experiment with same parameters, everything, still you may not get the similar result again. Yeah.
So would you recommend any particular software for conducting this type of studies?
For doing the molecular docking studies, one of the reliable software since long time, it's Autodoc. Autodoc is the reliable software from the Scripps Research Institute, where we can see some reproducibility of the results as well. Yeah, the software is somewhat validated and general public user software.
And would you like to comment on the various parameters which we need to keep in mind while doing the publication for these computational techniques?
What I would like to say to the all participants is that when you are trying to publish your idea, try to prove your idea through concept, and that means the proof of concept through experimental study as well. So your idea will be already validated and it is very reliable. So chances of continuing the research is very high. When you just simply produce a hypothetical imaginative conclusion or summary and publish in a research article, of course that can be carried forward by some other research team into the experimental study, can also extend their study to identify whether the concept published is proven experimentally or not. That's one way actually, because for example, your team members actually identified a hypothesis and published. So the other team who is actually having good backup, the funding and resources, So they may have considered your hypothesis and they do the experimental study. And once if they get success, of course, they can proceed further into the drug discovery pipeline as well. So what I would like to say, better to accelerate in the research process, of course, generating hypotheses. However, using reliable softwares are more recommended. Yeah.
So we have a question from participants P. Srinivasulu.
Mm.
So what is the success rate of drug discovery with these simulations?
Zero, it's actually 0 points. They say some reports they will say 0.02. Some publications they say 2%. Some publications I also referred 0.8 something like that. It's very less. Yeah. Unfortunately, because we are not actually addressing several parameters which are existing in the real environment. Several parameters we are not addressing in the computational simulations. But the parameter what we are considering in the computational simulation is comparatively bit more weightage. For example, several parameters are there. Okay, among the parameters, the parameter what we considered for doing simulation is relatively bit higher. That is the reason why at least some sort of success we have. For example, binding affinity is important. Binding affinity is due to binding interactions. So non-covalent interactions and hydrophobic interactions, everything. But there are other parameters like solvent effect and the multiple conformations. So many other things are there. But among the other things, the binding affinity is also important. For that reason, we are getting some sort of success. Yeah, that is the reason. That is the simple logic and rationale behind the success of the drug discovery. Why we are actually getting success means because the parameter what we selected to do the simulation is having some weightage. Yes.
So that was the same extension question from him that when the success rate is less than 0.5%, what is the need for conducting such kind of studies?
Oh, that's what I also addressed just now. Meaning to say that because the prioritization is possible, the prioritization is possible. What we say, because this is actually, as a medicinal chemist, if you ask me, I would prefer to do the experiment. I prefer to do the bioassay. I prefer to do the real experimental evidence. So what is this all about? The simulation is just to complement the process. Instead of doing 100 experiments, I may take one run of virtual screening and try to identify if any molecule is more potential binding. I start with that. Okay, molecule 59 is appeared to be potential binder. So let me start with 59. What's wrong? Because technology is available. Since technology is available, we can integrate the technology into practice so that if everything goes well, definitely we may end up with a new lead molecule at least. And one more thing, the success stories are clearly suggesting there are some chances to identify the molecule using computational simulations as well. And it all depends on the computational simulator capabilities and knowledge competencies, skill competencies, and understanding of the whole conceptualization. Yes, it is totally different. It is not simply as an end user we are doing some simulation, drawing some molecule and identifying the parameters using the software. It is not the actual thing. There are some additional things they will perform, meaning to say that they will apply the individual files. For example, Pfizer, how they prioritize the molecule? They prioritize the molecules based on Lipinski rule of 5. So it is a virtual screening process as well. And GSK, how do they prioritize the molecule? They prioritize the molecule based on Weber's rule. Weber's rule is also drug-like properties. So it all depends on what is the logical flow you are actually integrating into your research. So that logical flow may end up with success or may not end up with success because it is research. It's an explorative, not conclusive or decisive.
So I could not see any further questions. So with your kind permission, I will be closing the session.
Okay. Thank you all once again. Thank you, dear participants, for your time. And thank you all the organizers. Thank you, Dr. Pramod, for your graceful greetings and facilitating the section as a moderator. Thank you. See you all again. Bye.
Thank you, Vasudevara sir, for your elaborative presentation and for clarifying the doubts. So with this, we have came to the end of the sessions for day one of this refresher program training on pilot plant scale-up techniques. So on behalf of management and faculty of Chevrolet Hanma Institute of Pharmaceutical Sciences, we profoundly thank all the speakers and participants for their active participation. And again, we'll be meeting on day two sessions for tomorrow. Thank you all and have a great day.
Stay safe. Thank you.